Our next speaker is Kristine Coleman, who's the head of the Behavioral Sciences Services Unit at the Oregon National Primate Research Center. She's been in that position for about 12 years, and she ended up there after doing her PhD in behavioral ecology at Binghamton University and her postdoctoral work there at the Oregon Primate Center in behavioral neuroendocrinology. Her research interests are, of course, behavioral management, as well as individual differences in temperament and stress responsiveness. So without further ado, Kristine Coleman.
Okay, thanks Eric for inviting me to this conference and for organizing, and thanks Molly for such a great opening talk and explaining socialization and the problems we all face. So I'm going to talk today about some behavioral intervention and how socialization can be used as a behavioral intervention, and temperament, and we'll see how those go together later. As behavioral managers, we have two main overarching goals. One is to increase normal behavior for the animals in our care, and one is to decrease abnormal behavior. And as Molly mentioned, socialization is a really important part of this, of achieving these goals. Socialization increases species normal behaviors, including things like grooming, play, and Molly showed some great videos of some of these behaviors. And it also decreases abnormal behaviors. And again, as Molly mentioned, just recapping Molly's talk, some of the behaviors that socialization can help decrease include stereotypical behaviors, whole body stereotypical behaviors such as pacing, rocking, these behaviors that are repetitive without a seeming function, also self-directed behaviors, over grooming, self-hair plucking, eye poking, things like that, and also self-injurious behavior, self-biting, wounding, things along that nature. There are at least 3 ways in which socialization can affect the development and persistence of abnormal behavior. Past housing conditions can affect abnormal behavior. Whether or not animals were paired or socially housed in the past can affect their behavior in the present. Animals' current housing conditions can also affect their behavior. Whether animals are currently paired or social housed can affect whether or not they display abnormal behaviors. And socialization can be a form of behavioral intervention for animals that are showing behavioral problems. So I'll go through these. For past housing conditions, as Molly mentioned, past socialization really can affect many aspects of animals' lives, including whether or not they show abnormal behavior. There are several studies showing that single housing and length of time animals spent in single housing is correlated with abnormal behaviors. So animals that spent a lot of time in single housing tend to be more likely to show stereotypies, self-aggressive behavior, things like that later in life. And this is particularly true for younger animals. And so we know that inadequate socialization early in life is one of, if not the biggest risk factor for the development of self-injurious behavior and other behavioral problems later in life. So hand-rearing animals in a nursery without their mother is a huge risk factor for the development of abnormal behaviors later on. And as a result, our institution and many institutions are really reevaluating and refining nursery rearing techniques, how we handle infants that are abandoned or orphaned. At the Oregon National Primate Center, we've started a program that we call our fostering program for abandoned and infant orphans. And at the Oregon Center, just as a little background, we have about 4,500 rhesus macaques. Most of them live in large outdoor breeding groups, and we have about 12 to 1,300 indoor ones that live indoor in cages. And because we have a breeding group, periodically females either abandon their infants, they don't take care of them. Sometimes females get sick or die, and so we have orphaned infants. And when infants are abandoned or orphaned. In the past, we always brought them into the nursery and nursery reared them. In the last few years, we've started this fostering program in which we try to put these orphaned or abandoned infants with a foster dam. And a foster dam is just a dam. That's not the infant's biological dam. We first, of course, try a lactating female. That's very easy. Lactating females who maybe lost their own baby are often very receptive to new infants, and they care for them as their own, and that's relatively easy. But you can imagine we don't always have a steady supply of females who are lactating and recently lost an infant, and that's not a supply we would like to have. So we've started using some non-lactating females that are very baby-centered. They really like infants. But in order for these females who are very good mothers to act as foster infants, to act as foster dams to abandon infants, they have to be trained to allow the infants to feed. Otherwise, because they're good moms, they're very protective of the infants, and while they would rear the infants, they can't nourish the infants. So we've trained some of these non-lactating females to allow the infant to come up to a bottle and drink. And here this is kind of hard to see. It's through the bars, but you can use your imagination. There's an adult mom there and a baby drinking from a bottle there. And the female is being reinforced by the trainer for allowing the baby to come up and drink from the bottle. And we've had really good success with this program since 2009 when we started this. We've attempted to place 27 abandoned infants with these non-lactating foster mothers, and we've had a success rate of about 89%. So most of these animals do work. Early on, we looked at the growth rate of the infants. Our veterinary staff was very concerned that the infants were growing normally and at the same rate as an infant that was reared with in the nursery. And so this is just some preliminary or early work showing weight gain for about the first 3 months of life. And the purple bar there is the growth rate from nursery reared animals, and our foster infants grew at about the same rate. And to date, with our 24 infants, we've had no physical problems. We haven't had any infant that had to be pulled from the foster dam because it wasn't gaining weight or was sick or anything like that. We're continuing to look to see if this fostering program is having the intended behavioral effects. That is, we would like to see that these infants behave more like mother-reared infants than nursery-reared infants, and we're continuing to look at that, but we certainly think it will. So just to summarize, we believe that these foster dams can help reduce the need to hand-rear orphaned or abandoned infants, and that providing this early socialization with a mother, even if it's not the biological mother, will help reduce nursery rearing and will help increase socialization and ultimately help reduce the occurrence of abnormal behavior. And as I mentioned, we're continuing to look at this. So another way in which socialization can affect abnormal behavior is by the current housing conditions. And we know at our center, like I mentioned, most of our animals live in outdoor groups ranging in anywhere from about 30 individuals to about 200 individuals. And animals that live in large groups, at least at our center, have a much lower rate of showing abnormal behavior than animals in cages. So we know that present housing condition does affect whether or not an animal displays abnormal behavior. Daniel Gottlieb, who's a postdoc working with me, wanted to see if current housing conditions affected the presence of abnormal behaviors in our indoor house populations. So to do this, he used data from our behavioral assessments over a 10-year period from 2002 to 2012. And at our center, we do standardized behavioral assessments on all of our indoor animals on a regular basis, and we look for behavioral problems, stereotypy, alopecia, self-biting, things along those lines. We do a short temperament test. We do some other things as well. So he used those data. There was a total of over 4,000 adult rhesus macaques and over 11,000 observations, so it's a big data set. And He compared the type of housing, so in this case where they were full housed, grooming contact, or singly housed, and the behavior. And he specifically looked at whole body stereotypies, pacing, swinging, bouncing, those sorts of behaviors again, and alopecia. And importantly, he controlled for rearing history. So in the results I'm about to show you, rearing history was controlled for. At our center, animals can be housed in full pairs, and most of the time our animals are full paired. Molly showed some of our grooming contact bars earlier. That's our form of protected contact. This is, again, not the best photo to see, but there's a slide that has some bars that allow animals to put their hands through and groom each other. We do use this as a final housing method. Not as much now, but we did more earlier, closer in the 2000s. This graph shows the number of observations in which animals displayed stereotypies for the 3 different housing conditions. And as you can see, animals that were fully paired with another partner showed significantly less stereotypical behavior compared to animals in grooming or single grooming contact pairs or animals that were singly housed. And interestingly, we didn't see any difference in stereotypical behavior between animals in the grooming contact condition and the single housing condition. We saw the same trend with alopecia, and on the y-axis here is what we call our alopecia score, hair loss score, and that's a score that goes from 0 for a fully haired animal to 5 for a fully bald animal. So the lower numbers, the more hair the animal has, and animals that were in full contact had better hair coats and less alopecia, again, than those in grooming contact or those that were singly housed. And so this fits with the study Molly described earlier, that there seems to be, for rhesus, a difference between fully paired and grooming contact paired animals, and at least with respect to these measures in our study, the animals in grooming contact seem to behave more similarly to those that were singly housed compared to those that were fully paired. Daniel did a similar study when he was at the Davis National Primate Center and had similar results. So these have been found elsewhere as well. I'm going to switch now and talk about how we can use socialization as a behavioral intervention. And we've probably all heard of Harry Harlow and his work early in the 60s and 70s. He did a lot of rearing of rhesus macaque infants, and he reared them in isolation and found, not surprisingly, that they had, not surprising now, that they developed a lot of abnormal behaviors. And early work in the 70s by Harlow and his students, Steve Sumi and Melinda Novak, showed that they could reverse some of these abnormal behaviors by pairing these isolated infants that were isolated from birth, with animals that were significantly younger. When they tried pairing them with age-matched conspecifics, the isolates tended to be the victim of some aggression, so that didn't work well for them, but they were able to reverse some of the abnormal behaviors by pairing them with a less socially mature animal or a significantly younger animal. It didn't reverse all of the abnormal behaviors, but it did give an insight that socialization can help reduce abnormal behaviors. Of course, we don't rear infants in isolation anymore, at least I hope nobody does that. But there are still several studies that have shown that social housing can be a behavioral intervention for animals that do show behavioral problems. And one example from Jim Weed, here the NIH's own Jim Weed, he used pairing as a way to help reduce self-injurious behavior in 6 male rhesus macaques. These monkeys were all showing self-injurious behavior. They were vasectomized so that they could be paired with a female, and 5 out of the 6 animals did show a reduction in self-injurious behavior. So it seems that pairing can be used in some circumstances to help reduce abnormal behavior for those that have it. In addition to helping reduce abnormal behavior, social partners can also help reduce stress and the effects of stress. In a study that Kate Baker did, they showed that rhesus macaques that were paired after having been singly housed were less responsive to stressful stimuli when they were pair housed compared to when they were singly housed. So pair housing seems to help reduce abnormal behaviors associated with stress. And in addition to abnormal behaviors, stress can affect many other parameters as well. It can affect physiology and health of the animals. So the social companions can help mitigate the effects of stress and then presumably help reduce abnormal behaviors as well as improve health. We're looking at this again at our center. This is we call it a satellite cage. This is a group of animals of about 40 individuals. One of the females, this girl right here, had some mastitis and needed to be pulled from the group. Typically, we would put the animal in the hospital, but then she would be away from her social group. So we use these satellite cages so that the animals can stay in visual contact with the rest of their group. And we're looking into whether this helps them with speedier recovery and helps reduce the stress associated with being sick and being removed from your group. So to summarize, socialization is an important to the behavioral health of non-human primates. It's an important part of behavioral management plans. Appropriate socialization can help the development of abnormal behaviors, and socialization can be an effective intervention for behavioral problems. It can also help mitigate the effects of stress. However, there are some exceptions. It doesn't always reduce the occurrence of abnormal behavior. There was a study that showed that after pairing, animals did show an initial decrease of abnormal behavior, but after the pairs were well established and the animals were more familiar with each other, abnormal behavior started to increase again. And anecdotally, we've had examples at our center of animals that exhibited self-biting behavior. We put them into a group, and the animals continue to self-bite. So while it is certainly worth trying to socialize animals with behavioral problems to see if that helps reduce the behavioral problem, of course, you still need to monitor the animals, and some animals do need treatments other than socialization, pharmacological interventions and other sorts of treatments. And social housing can cause abnormal behavior or exacerbate abnormal behavior if the partners aren't compatible. So compatibility, again, as Molly mentioned, is a really important part of socialization. And that, well, now we'll switch gears and talk a little bit more about compatibility and how we can find compatible partners for the animals we're trying to socially house. Compatibility can be influenced by many different factors. There are species differences. We have mostly rhesus macaques at our center, but we do have some baboons, and again, anecdotally, they apparently are much easier to pair house compared to our rhesus. I think just about anything is easier to pair house compared to rhesus. Age can play a factor. Sex can play a factor. I'm going to show a couple of graphs of some statistics from the Oregon Primate Center. These are our pairing success, and these are numbers taken from about 1,400 pair attempts that we've made since 2011. And we've defined success as animals that stayed together, didn't need to be removed for fighting or aggression for at least 2 weeks, and a non-successful pair would be one that started fighting immediately. We had about 80% of our adult animal pairs that involved 2 adults, about 80% were successful. For juveniles, and that's any animals between 1 and 3 years of age, we had close to 100% success rate. For our aged animals or pairs in which one partner was at least 15 years of age, our success rate was more like 56%. And these numbers are, I think, a little bit lower, but very similar trend to what Molly showed a little bit ago. And we've begun to put adult animals with juveniles, especially animals that are very hard to, that we've had limited success pairing in the past, or animals that have been in singly housed conditions for long periods of time. We find that they a lot of times will do better with a younger animal rather than somebody around their own age. And we have about 97% success rate pairing an adult with a juvenile. And these aren't related. It's not, you know, mom and her daughter or something like that. Similarly, if we look at pairing success and sex, we have, again, about an 80% success rate for our female-female pairs. We have about a 90% success rate for our male-male pairs. So again, this, as Molly showed, we have better success pairing our males than our females. And we have about a 93% success rate pairing our males and females. And the number in parentheses are the number of attempts we tried for that particular group. There are some other factors that can influence animals' compatibility, including past experience, especially past social experience. And as Molly showed, animals that were singly housed for long periods of time can be more challenging to pair than animals that have a rich social history, especially animals that came from a social groups, things like that. And another factor that I'm going to spend some time now talking about is personality or temperament. So I'll start with, what is temperament? Sidetrack a little. Temperament and personality are often used interchangeably. It's not always been the case, but now people refer to temperament and personality to mean basically the same thing. And it, in essence, refers to an individual's stance towards environmental change. How do they deal with stressful and novel stimuli or experiences? Temperament's been shown to emerge early in life and remains relatively consistent throughout development. There are a number of ways in which personality and temperament can be assessed. They're often questionnaires. They're not actually given to the animals, but usually to the caretakers who are asked about to describe the animal's personality. Are they fearful? Are they shy? Are they bold? Animal caretakers tend to know their animals pretty well, and so this can be an effective way to assess temperament. A lot of investigators use a rating system, watching the animals in their home environment, and they rate them on traits such as curiosity, boldness, aggressiveness, things like that, and then these variables are usually put into a principal component analysis or some sort of factor analysis, and different factors emerge that deal with personality. Another way that temperament can be assessed is by assessing the animal's response to challenge, and this is used a lot in animal studies, non-primate animal studies, to assess temperament. One axis of temperament that I'm really going to focus on, so I'm going to say temperament, but what I really mean is an axis of temperament from behavioral inhibition to exploration. And this axis is one that's often measured by response to novel stimuli. So individuals who in these tests, individuals are presented with a novel object, a novel conspecific, a novel environment, something novel, and their responses are quantified. And you'll see, as I'm sure if you work with monkeys or animals or anything really, you know that animals show a lot of individual differences on this axis. Some individuals respond to novelty by going and inspecting it, and they tend to almost seek out novelty. These individuals might be called exploratory or bold or even risk takers, depending on the study. At the other end of the extreme are animals who want nothing to do with novelty. They shy away from it. In studies in children, they often start crying when presented with novel objects or novel people. These individuals might be called inhibited or shy or risk averse, and of course there's a bell-shaped curve in between. This axis of from behavioral inhibition to exploration is one of the most stable temperamental axes, and it's conserved across species. So pretty much every species in which this trait has been looked for, it's been found. There are shy and bold monkeys, of course, people, of course, but also this individual differences on this trait have been found in dogs and foxes and fish, hermit crabs, octopus, so a wide variety of species. So it just points to its importance that it has been conserved across all these species. So we wanted to, because my background is in individual differences in temperament and I'm interested in it, we a few years ago did a couple of studies looking to see whether temperament could help us predict pair compatibility, whether this was something that could help us make better pairs. And we did a couple of studies. In our first study, we looked at 19 female rhesus macaques, adults, each who had had one successful and one unsuccessful pairing attempt. And again, a successful pairing attempt is one in which the animals stayed together for at least 2 weeks without major fights. And an unsuccessful pairing attempt was one in which the animals started fighting immediately and we didn't proceed with that pair. We then measured temperament in all 57 animals, so the 19 focal individuals, the successful and unsuccessful partner, and we measured temperament using a standard novel object test. And in our novel object test, we presented the animals with 2 novel objects. One was a novel food item, a pretzel, which we placed in the monkey's cage, and I don't know what it is about Oregon monkeys and pretzels, but a lot of our monkeys are very fearful of pretzels. So, not sure what to make of that, but they are. We then observed the monkeys for about 5 minutes, and we recorded their latency to inspect, touch, and then eat the pretzel. We then hung a colorful bird toy that was novel to the animals on the outside of their cage, and again watched them for 5 minutes and recorded their latency to inspect, touch, and manipulate the toy. And I guess a picture is worth 1,000 words, so here you go. This monkey here, we might call exploratory. She went right up, started inspecting the bird toy hung on her cage, whereas this one stayed in the back, really wanted nothing to do with that brightly colored bird toy, and she would be an example of a more inhibited individual. After we conducted the temperament test on all these monkeys, we gave them an inhibition score based on their latency to inspect, touch, and manipulate the 2 objects. And the scores went from 0 to 6. A score of 0 meant that the monkey immediately explored both of the objects, and a score of 6 meant that the monkey never explored either object. And the scores in between meant that maybe they explored one of the objects and not the other, or took longer to do so. We then calculated the difference in inhibition scores between the subject monkey, the focal individual, and her successful partner, and the subject monkey and her unsuccessful partner to see if there was a difference. And here results on the y-axis is the inhibition score difference, so the difference in the inhibition scores between the 2 monkeys. An inhibition score difference of 0 means that they had exactly the same inhibition score. In other words, their temperament was very similar. And an inhibition score difference of 6 meant that one animal had a score of 0 and one animal had a score of 6. And so they were as opposite as you could get. And this bar is the difference in inhibition scores between the monkey and her successful partner. Here is the difference between the monkey and her unsuccessful partner. And as you can see, the animals were more similar in temperament with their successful partner compared to their unsuccessful partner. We then had a unique opportunity at our center. We were getting a shipment of about 74 female rhesus macaques, and we thought this would be a very good opportunity to test out this study in a prospective kind of, or to test out our hypothesis in a prospective way. So we assessed temperament using the same temperament tests in all 74 adult female rhesus macaques. We categorized them based on their inhibition scores. So an animal that got a score of 1 or 2, we called exploratory. An animal that got a score of 5 or 6, we called inhibited. And the others we called moderate. And then we made pairs based on these categorizations. So we attempted 18 pairs in which the animals had the same temperament. So we paired an inhibited animal with an inhibited animal, say, or an exploratory animal with an exploratory animal. And then, and the other 19 pairs had different temperaments. So maybe an exploratory animal with a moderate animal or something a lot like that. And in this particular case, most of the pairs were successful, which was very good for the pairs and somewhat challenging from the research part of it. But only 3 of the pairs were immediately unsuccessful. We only saw fighting in 3 of the pairs. So we decided to then look at the behavior of the pair. And so we conducted focal observations on the pairs, and we looked for behaviors like close social contact, grooming, huddling, things like that, proximity, sitting close to another individual, non-social, the situation in which animals are on opposite sides of the caging, not really interacting with each other at all, or aggressive. And we decided to look at the behavior because we had been calling a pair successful simply because they didn't fight, but we wanted to see whether or not they were showing positive pro-social behaviors. This graph shows a histogram of the amount of time the pair spent in close social contact. So you can see there was a lot of variation. Some animals spent most of their time in close social contact, while others spent very little time in close social contact. So we then categorized the pairs into good pairs, and those were pairs that spent at least 50% of the focal observations in close social contact, and incompatible pairs, and those were pairs that either were immediately unsuccessful, so the 3 that fought immediately, or pairs that in which the animals spent less than 25% of their time in close social contact and had some sort of aggressive interaction, some biting, hitting, things like that. And then we compared the inhibition score differences between our good pairs and our not quite as good pairs. And we found that, as before, the pairs that were good pairs, the pairs that showed a lot of close social contact, had more similar temperaments compared to the pairs that showed very little close social contact and showed some aggression. So pairs consisting of animals with more similar temperaments were more compatible than those with different temperaments. And these results were similar to a study that John Capitanio and his student found. They were looking at animals in large social groups, but what they found in their study was that animals tended to be close or hang out, say, with animals of similar temperament compared to animals with different temperaments. So temperament is one way, along with all the other factors we've mentioned, things like age and past social experience, to help guide our choices about potentially compatible individuals. And went faster than I thought, so I just want to thank the people who helped do these studies and who take such good care of the animals out in Oregon. Are there any questions?